Ladies and gentlemen, thank you for this opportunity to present, and thank you for choosing to listen to this presentation at the IHPBA 2020 on demand. The liver is remarkable and unique for its ability to undergo major surgery to remove cancer and regenerate afterwards. And advances in liver surgery have made cure of advanced cancer possible for many people. The ability of the liver to withstand and recover from major surgery depends on three main properties: the future liver remnant volume, the health and function of the liver remnant, and the capacity of that liver to regenerate. Together, we call this the future liver performance. In this project, we have built a non-invasive, quantitative MRI algorithm that is able to provide information about liver health before surgery in an anatomically discrete way that will lead to safer liver surgery planning. Here you can see many examples of the technology in action. Using false colour, corrected T1 is red, often mapping to fat, so you can see a small tumour in a very unhealthy fatty liver here, and in the top left corner, a fatty hepatocellular carcinoma in a cirrhotic liver, whereas on the right you can see a more fibrotic colorectal liver metastasis in a relatively healthy looking liver. For an individual considering major liver resection, this preoperative information may be life-saving. In the top example, a large tumour burden may still be resectable if we know that the future liver performance will carry that individual patient through, whereas in the bottom example, even a minor set of atypical resections may be too much for this very unhealthy liver. So we can use this information to make surgical decisions, but also counsel each individual about their risk, a real-world application of personalised medicine. For this individual, we may push the boundaries to achieve tumour clearance, whereas for the individual below, we might be more reluctant to operate and direct the patient towards alternatives, for example, ablation. Our aim in this project was to develop and test Hepatica for its ability to forecast future liver performance in patients undergoing liver resection. We built a team from around the UK with expert liver surgery centres in Edinburgh and in Basingstoke, and teamed up with Prospective Diagnostics, who developed the technology, and with our funders Innovate UK and with North Hampshire Medical Fund. Hepatica is an adaptation of liver multiscan, a quantitative MRI algorithm that is non-invasive, non-contrast, rapid, and can be done whenever a patient attends for a usual liver MRI. We scanned individuals before surgery and again 6 weeks after surgery, and in a nested cohort we scanned patients at recruitment, after neoadjuvant chemotherapy or portal vein embolization, and again immediately prior to surgery, and again in the post-operative phase. Our primary outcome was a correlation between the Hepatica score and a modification of the Hyder-Paulick score. The Hepatica score is a corrected CT1 index weighted by future liver remnant volume that was statistically derived using a generalized linear model. We modified the Hyder-Paulick score, previously published, to use the integral of all scores from day of surgery to day 5 rather than just the day 3 in the original score, which, as you will see later, was the correct choice when you can see the individualised data plotted. We approached 419 individuals considering liver surgery, of whom 305 met the criteria and 149 participated. Just under half of patients had major resections, a similar proportion had neoadjuvant chemotherapy as shown, and the majority of resections by far were for colorectal liver metastases. Patients' liver health preoperatively followed a normal distribution with a skew to the unhealthy end of the distribution. As you can see, the peak time for liver dysfunction is on post-operative day 2, and you can see that the integral, or area under the curve, gives a much more sensitive readout of post-operative liver dysfunction than any single time point measure might. Hepatica performed well to predict liver dysfunction, with an area under the receiver-operator curve of 0.76. Additional statistical outcomes are shown on the top right-hand corner of this slide. A thresholded Hepatica score above 800 was also associated with longer inpatient stay, and so Hepatica has a direct healthcare resource use outcome. The liver multiscan itself performed well for fat and iron, and to a lesser extent for inflammation. In practical terms, therefore, the individual on the top of the slide had a healthy Hepatica scan and had a low amount of liver dysfunction and an in-hospital stay of 6 days despite a relatively major resection, whereas this individual with an unhealthy liver had a significant amount of liver dysfunction and stayed in for 14 days despite a relatively minor liver resection. But why is this technology any better, for example, than using indocyanine green, ICG, or mebrofenin, or just future liver remnant volume? Well, because for the first time we can actually map future liver performance of an individual liver segment to the liver segmental anatomy. And we know from the scans and generally that liver disease itself does not affect all segments equally. To automate this, we applied machine learning and artificial intelligence to identify intrahepatic structures, and then we could use that to map the Couinaud segments, thus giving segment and volume a functional score, so that the accuracy of predicting future liver performance is based on the actual segments that are being left behind, not just on the preoperative performance of the whole liver. Finally, to return to liver health after surgery, the liver needs to regenerate. And what we see here is a tantalizing insight into the health of the regenerated liver. Here you can see on the right that the regenerated segment of the remnant liver, the health of the remnant liver improves after regeneration. And that follows through into the overall cohort. Now, this may seem intuitive at first, but when you think about it, the implications of this finding are that it might actually be beneficial to resect more parenchyma in a less healthy liver to achieve, eventually, after regeneration, overall a better outcome. Now, this is a really controversial proposition, but it is worth thinking about. Lastly, we can start to unpick some of the biological mechanisms that underpin these observations. For example, using metabolomics, where here we have identified key metabolic pathways that are associated with a better or worse liver regeneration. In summary, therefore, Hepatica is a feasible, non-invasive, non-contrast imaging technology that adds valuable information to assist the decision-making and planning around surgical liver resection. Hepatica may add reassurance to allow extended resections, and it may add caution in the case of less healthy livers. What it will do is it definitely informs the risk discussion with the patient and will increase safe decision-making and adds to our overall scientific knowledge. Thank you for choosing to listen to this talk, and thank you to all the team and all our collaborators.